there! Welcome to Merivit Education Center, and this is Science with Master Rick. In today's lesson, we will be learning part 2 of cell division, which is meiosis. Meiosis is the process in eukaryotic sexually reproducing animals that reduces the number of chromosomes in the cell before reproduction. Now, many organisms package these cells into gametes, such as egg and sperm. The gametes can then meet during reproduction and fuse to create a new zygote. Because the number of alleles are reduced during meiosis, the combination of two gametes will yield a zygote with the same number of alleles as the parents. Meiosis is necessary for many sexually reproducing animals to ensure the same number of chromosomes in the offspring as in the parents. The act of fertilization includes two cells fusing together to become a new zygote. If the number of alleles of each gene is not reduced to 1 in the gametes that produce the zygote, there will be 4 copies of each gene in the offspring. In many animals, this would lead to many developmental defects. Meiosis then consists of 2 cell divisions, known as meiosis 1 and meiosis 2. In the first division, which consists of different phases, the duplicated DNA is separated into daughter cells. In the next division, which immediately follows the first, the 2 alleles of each gene are separated into individual cells. The following are description of the 2 divisions and the various phases of stages of each meiosis. Remember, before meiosis starts, the normally diploid DNA has been duplicated. This means there are now 4 copies of each gene present in 2 full sets of DNA, each set having 2 alleles. Meiosis is preceded by an interphase consisting of the G1, S, and G2 phases, which are nearly identical to the phases preceding mitosis. The G1 phase, which is also called the first gap phase, is the first phase of the interphase and is focused on cell growth. The S phase is the second phase of interphase, during which the DNA of the chromosomes is replicated. Now finally, the G2 phase, also called the second gap phase, is the third and final phase of interphase. Now in this phase, the cell undergoes the final preparation for meiosis. During DNA duplication in the S phase, each chromosome is replicated to produce 2 identical copies called sister chromatids that are held together at the centromere by cohesin proteins. Cohesin holds the chromatids together until Anaphase 2. The centrosomes, which are the structures that organize the microtubules of the meiotic spindle, also replicate. Now this prepares the cell to enter prophase 1, the first meiotic phase. Prophase 1 is the first step in meiosis 1. It is similar to prophase in mitosis in that the chromosomes condense and move towards the middle of the cell. The nuclear envelope degrades, which allows the microtubules originating from the centrioles on either side of the cell to attach to the kinetochores in the centromeres of each chromosome. Unlike in mitosis, the chromosomes pair with their homologous partner. Now this step does not take place in mitosis. At the end of prophase 1 and at the beginning of metaphase 1, homologous chromosomes are printed for crossing over. Between prophase 1 and metaphase 1, homologous chromosomes can swap parts of themselves that house the same genes. This is called crossing over and is responsible for the other law of genetics, the law of independent assortment. Now, this law states that traits are inherited independently of each other for traits on different chromosomes. This is certainly true all of the time. For traits on the same chromosome, crossing over makes it possible for the maternal and paternal DNA to recombine, allowing traits to be inherited in an almost infinite number of ways. In metaphase 1 and meiosis 1, the homologous pairs of chromosomes line up on the metaphase plate near the center of the cell. This step is referred to as a reductional division. The homologous chromosomes that contain the two different alleles for each gene are lined up to be separated. The chromosomes line up on the metaphase plate with their homologous pair. There is no order upon which side the maternal or paternal chromosomes line up. This process is the molecular reason behind the law of segregation. The law of segregation tells us that each allele has the same chance of being passed on to an offspring. In metaphase 1 of meiosis, the alleles are separated, allowing for this phenomenon to happen. Much like anaphase of mitosis, the chromosomes are now pulled towards the centrioles of each side of the cell. However, the centrosomes holding the sister chromatids together do not dissolve in anaphase 1 of meiosis, meaning the only homologous chromosomes are separated, not sister chromatids. In telophase 1, the chromosomes are pulled completely apart and new nuclear envelopes form. The plasma membrane is separated by cytokinesis and the two new daughter cells are effectively formed. The two new cells, each haploid in their DNA, but with two copies, are the result of meiosis 1. Again, although there are two alleles for each gene, they are on sister chromatid copies of each other. These are therefore considered haploid cells. These cells take a short rest before the second division of meiosis, meiosis 2. Prophase 2 resembles prophase 1. The nuclear envelope disappear and centrioles are formed. Microtubules extend across the cell to connect to the kinetochores of individual chromatids, connected by centromeres. The chromosomes begin to get pulled toward the metaphase plate. Now, resembling mitosis, the chromosomes line up with their centromeres on the metaphase plate. One sister chromatid is on each side of the metaphase plate. At this stage, the centromeres are still attached by the P protein cohesin. Cohesin is a protein complex that mediates sister chromatid cohesion, homologous recombination, and DNA looping.
The sister chromatids separate. They are now called sister chromosomes and are pulled towards the centrioles. This separation marks the final division of the DNA. Now, unlike the first division, this division is known as an equational division because each cell ends up with the same quantity of chromosomes as when the division started, but with no copies. As in previous telophase 1, the cell is now divided into two, and the chromosomes are on opposite ends of the cell. Cytokinesis, or plasmid division, occurs, and new nuclear envelopes are formed around the chromosomes. At the end of meiosis 2, there are 4 cells, each haploid, and each with only one copy of the genome. These cells can now be developed into gametes, eggs in females, and sperms in males. Mitosis and meiosis are the two ways by which cells reproduce. As a result, they share several steps in their respective processes. Meiosis adds another division and a step that mixes the genetic material from the parent cells. But in both cases, the cell must duplicate its DNA, pull it apart in two sets, place the sets on each end of itself, and then divide down the middle. Both mitosis and meiosis produce fresh new cells based on their parent's cell's genes. Mitosis produces two cells from one parent using one division event. But meiosis, on the other hand, produces four new child cells with two divisions, each of which has half the genetic material of its parent. Now, mitosis takes place all over the body, while meiosis, it only takes place in the sex organs and produces sex cells.
And that ends our topic for part 2 of cell division in meiosis. And as always, remember, science is cool.